Good morning, good afternoon, and good evening, everyone, to wherever you are in the world today. I'm Mark Leefring, the Science Advisor at the Ag Emission Centre, formerly known as the New Zealand Agricultural Greenhouse Gas Research Centre, and we are delighted to welcome you to today's webinar. This State of the Science webinar series is hosted by the Ag Emission Centre, and today's webinar is in partnership with Teagasc Climate Centre in Ireland. These webinars ensure that you have access to some of the best local and global experts when it comes to agricultural greenhouse gases. Please note we have a number of exciting webinars planned throughout next year, so keep an eye out on our social channels for more information. It's a great lineup today, and we're also lucky to be joined by Carl Richards, the head of the Teagasc Climate Centre, who will be your chair and take you through today's sessions. Welcome, Carl, and over to you.
Thanks, Mark, and good morning and good evening, everybody. So, look, you're very welcome to today's webinar, which is the next in our series of joint webinars. We're being joined by Cecile de Klein, who's the principal scientist with the Ag Research Group of the New Zealand Bioeconomy Science Institute. And she has more than 30 years research experience in nitrogen cycling and greenhouse gas emissions from grazed livestock systems. She leads the low greenhouse gas plants program from the Ag Emission Centre on evaluating the potential of plantain to reduce nitrous oxide emissions from urine patches. Cecile also works closely with policymakers and the agricultural sector to refine national inventories as well as on-farm accounting models. We're also joined by Dominika Kroll from Teagasc, who is a senior research officer at the Teagasc Crops, Environment and Land Use Programme based in Johnstown Castle. Dominika is also the lead for the nitrogen research pillar within the Teagasc Climate Centre. Her research interests include the impact of management of organic manures, synthetic fertilizers on emissions of nitrous oxide and ammonia from grassland systems. She is contributing to quantification and mitigation of these emissions and refinement of national inventories. So we've got two very prestigious speakers this morning who are going to give us, I suppose, the latest science, both in New Zealand and across Ireland. So I'm going to hand over now to Cecile, who will give the first part of the webinar.
Thank you, Carl, and kia ora tato, and thank you for the opportunity to share with you our experiences on the low greenhouse gas plant program that the Ag Emission Centre has funded over the last few years. I'll start sharing my screen. Does that come through okay? Yep. Okay, thanks for the thumbs up, Carl. So the title of my talk is a question. Can plantain reduce N2O emissions from pasture-based livestock systems? And spoiler alert, the answer is we don't know just yet. But as you'll hopefully see throughout my talk, there is potential, and there are good opportunities. So before I kick in, I really would like to, of course, thank my co-authors listed here who've done a lot of the work that I'll be presenting on their behalf. And I also would like to acknowledge all the technical staff that are doing all the enormous amount of gas measurements done during the experimental work. Okay, and then before I really kick in, I'm not sure where everybody's joining from, but some people may think of plantain as something different that we are thinking of. So in this talk, plantain is Plantago lanceolata as a pasture species and not the fruit plantain that some of the audience may think it is. So just as a little bit of background in terms of New Zealand agriculture and also our greenhouse gas emissions, our New Zealand agriculture is a really important part of our economy. And as a result, it contributes quite significantly to our greenhouse gas emissions. About half of our emissions are coming from agriculture. And because it's largely agriculture is dominated by grazed livestock systems, a lot of those agricultural emissions are methane from enteric fermentation of the animals, and about 20% of this is nitrous oxide from soil. And our systems are predominantly pasture-based grass clover pastures, and that are animals that are grazed virtually year-round, if not all year, by the grazing animal, and the urine patches that are deposited by these animals are the key sources of our nitrogen losses and nitrous oxide, and also in leaching. And in recent years, plantain has been identified or shown, suggested as a good option for helping to reduce those nitrogen losses. And the way plantain does that, there's various options available, or various mechanisms that are being proposed that plantain can reduce nitrous oxide emissions. And the first two that are listed here is what I call the animal or urine effects. So the first one is about that plantain in the diet of the animal can reduce nitrogen loading rates in the urine patch, and as a result reduce emissions because there's less of the nitrogen going into the urine patch. I won't be talking about this mechanism much today. The second mechanism, this animal mechanism, if you like, is where plantain compounds that are in the plantain plant that are consumed by the animals can come through the animal and be excreted in the urine, and those compounds, we call them BNI compounds, or biological nitrification inhibitors. And these inhibitors can prevent the conversion of ammonium into nitrate, and nitrate is the precursor of nitrate leaching as well as nitrous oxide emissions. So by inhibiting that nitrification process, nitrogen losses can be reduced. Those compounds can also get into the plant through a third mechanism, or into the soil through a third mechanism, and that is where plant roots, the plantain roots itself, exude those compounds into the soil. And then lastly, another plant effect is really that the plantain plant, through its morphological differences compared to, say, ryegrass, above ground as well as the root compounds can be very different, can affect the soil microclimate and the nitrogen cycling as well, through changes in differences in evapotranspiration, or causing shading, or having differences in uptake. So those are four mechanisms really by which plantain can affect the nitrogen cycle. So the aim of the program that the Ag Emission Center funded for us was really to try and quantify the potential of plantain to reduce nitrous oxide emissions from urine patches, but then secondly, also identify what are the key factors that are affecting that reduction capacity of plantain. We used various approaches to do this, to test this, and we had field and lysimeter experiments where we looked at different proportions of plantain in the sward and applied urine. Livestock urine was applied to those swards. And in particular, our lysimeter studies, they were in collaboration with Dairy NZ Plantain Potency and Practice Program, and we're very grateful to that program to be able to piggyback, if you like, onto their lysimeters and measure nitrous oxide emissions from them. We also conducted quite a few glasshouse experiments where we used soil mesocosms, we call them. So basically just buckets with monocultures of ryegrass and plantain. And we looked at the effect of the soil nitrogen status and also soil moisture content on plantain's ability to reduce nitrous oxide emissions. And finally, we did some quantitative analysis on the data that we've collected over the years to try and really identify what can we distill, what are the key factors that are driving the variability in what we see in our field results. So one of our first field studies we conducted was down here in the south where I'm based, and these were large plots that were sown with different proportions of plantain. We had here, these results show 4 proportions. I think in the end we had 6 proportions, but this part of the trial had 4 proportions of plantain. And we measured nitrous oxide emissions following application of cattle urine that was collected from animals that were grazing standard pasture. So there was no plantain in the diet. So the effect we're seeing here is purely the plant effect, so the sward effect. So these swards were about 4 months old when we applied the urine, and you can see on the bottom of the x-axis the nominal proportion of the sowing rate of plantain in the sward, and on the y-axis the emission factor of nitrous oxide, which is the amount of N2O that was emitted as a proportion of the N that was applied. And As you can see, there was a significant and reduction in nitrous oxide emissions, a trend following with increasing proportion of plantain, and up to a 40% reduction in nitrous oxide emissions we found. So plantain can reduce emissions, but the results are also quite variable. So these results are from a lysimeter study, one of our first lysimeter studies, again with swards, with different proportions of plantain and receiving cattle urine collected from animals that were grazing grass clover pasture. So again, no plantain in the diet. So this is again the plant effect. So mechanism 3 and 4 that I talked about earlier. And here we see the results are quite different to what we found in the field study, very variable. And as you can see in the allophanic soil, it's a free draining soil in the north of New Zealand. The biggest reductions were found when we had 30% plantain, nominal sowing rate of plantain. And in the clay soil, quite a heavy soil, there was no effect, no significant difference between the different plantain proportions. I said nominal proportion of plantain because when the team actually looked at what was actually in the proportions in plantain, they compared the above ground dry matter in terms of what was actually in the plantain versus the cumulative N2O emissions. For the allophanic soil, they actually found there was a reduction, a significant reduction, if you look at what was the actual plantain proportion in the sward. We didn't find that in the clay sward, so again, some uncertainty around why that would be. Although the emissions were quite high, it was wet here, and the clay soil is a heavy soil, was quite moist. So we think some other soil processes were happening there. So a couple of years later, new lysimeters were taken from exactly the same paddock where the first lysimeter was taken from. And again, allophanic and clay soil with nominal proportions of plantain, the same as in the first study, but there was no significant difference there in the allophanic soil, and there was some variability in the clay soil with 50% portions of plantain, quite high emissions. Note that the error bars are very different here, or the y-axis scales are different here. So the emissions in that second study were much higher than in the first study. So that could also have affected the outcome. In the most recent lysimeter study, N2O emissions were again affected by pasture type. So here we have explanation, or you show the first year, 1-year-old swards where urine again was applied from ryegrass urine. So again, urine that was taken from animals grazing ryegrass white clover, so no plantain in the diet. And there was a significant reduction of nitrous oxide emissions when that urine was applied either to a mixture, which had about 50% of plantain in the sward, or with a pure plantain sward. In this study, we also looked at mechanism 2, if you like, if you can remember what that was, but that was really there where compounds of the urine or compounds of plantain that were ingested by the animal were then returned to the soil in the urine. So we tested this. So in the orange bar, you see where the urine that was applied was taken from animals that were grazing the mixed sward. And the green bars are showing the results for animals that were grazing the plantain sward, and the urine collected from there was all at the same nitrogen content. The nitrogen content was normalized, but this was testing whether there were compounds that were ingested through the plantain and coming out with the urine into the soil. But as you can see, if you compare the first blue with the first orange and the first green bar, there's no significant differences if those different types of urine were applied to the ryegrass sward, or if they were applied to the mixture or through the plantain swards. So these results are really showing that the results we're finding here is that plantain can reduce N2O emissions compared that there is a sward type effect or a pasture type effect, so a plant effect, but there was no differences among the urine sources. So in this case, we couldn't really confirm that mechanism 2 was happening in this case. So the next study we looked at was really trying to delve into a little bit more of the detail of how plantain could reduce emissions. And here's a mesocosm trial, a glasshouse study, where we looked at the hypothesis that the capacity of plantain to reduce N2 emission is highest when there is low nitrogen in the soil. Because the theory is that plantain exudes the compounds that might inhibit nitrification if there is little nitrogen available. So they conserve the nitrogen in the ammonium form, which is more easily accessible for plants and is less mobile and doesn't leach or doesn't get lost as easily. But so we had 3 soil nitrogen status mesocosms that we used, and we had grown 100% ryegrass or 100% plantain, so monocultures of the 2 species, and those buckets with the soil with different nitrogen contents. And we found the opposite of what our hypothesis was, because if our hypothesis was true, we would have seen that the red bars would have been the same, and the green bars would have been maybe a small difference, where the biggest difference we would have expected between the blue bars. But we actually found that that is not the case. So even if there's a high N content in the soil, plantain seems to be reducing the nitrous oxide emissions. Another mesocosm study that we did, we looked at what actually the effect of soil moisture content was. So here you see the results of mesocosm study where we had different soil water filled pore space induced in the plants, in the buckets. We had monocultures of ryegrass and plantain again, and we see that there is, particularly under the higher soil water filled pore spaces, there is a reduction in plantain. Didn't see an effect in the 30%, which is a very dry soil, so there's very low emissions to start off with. So plantain didn't have an effect there. But as soon as you get above 50% water filled pore space, there was a reduction. And the overall effect of these treatments was a 22% reduction in nitrous oxide emissions under the plantain monoculture. In this study, we also looked at what happened with the nitrogen in the system and the plant uptake. And it actually showed that plantain took up more nitrogen out of the system in the monocultures of plantain. So this could be one of the reasons why nitrous oxide emissions were potentially lower, because it didn't affect, it take away the nitrogen in the soil, and therefore there's less available for nitrous oxide emissions. So a significant increase in nitrogen uptake of about, on average, 15% under the plantain sward. So as I mentioned before, we also looked at all our data that we had available to date, and then a meta or a data analysis, quantitative analysis of this data. It's a little bit of a busy slide, and I'll take you through it. But on the left-hand side, you see where we organize the data by urine application rate, and either that was 400 to 600 or larger than 600. And on the y-axis, there's the log emission factors. So the data presented on a log scale, but these are the N2O emission factors. And what we saw here is that that when the urine rate was relatively low, between 400 and 600, plantain swards, the red boxes, had significantly lower emissions compared to ryegrass swards. And similarly, for when we looked at soil field capacity, so the physical conditions of the soil, that where there was a high soil field capacity, so the soil could hold a larger amount of water under field capacity conditions, there was a significant effect of plantain on nitrous oxide emissions, but not when the soil field capacity was lower. So those 2 factors seem to be explaining some of the variability that we're finding. And finally, the really thing that I would like to point out here is that we don't really know what happens, what plantain does to increase complete denitrification. And with complete denitrification, I mean is that this is a process in the soil that where nitrous oxide, before it is emitted into the atmosphere, can actually be utilized by microbacteria to be reduced to benign, environmentally benign nitrogen gas. And that is the only known sink of nitrous oxide in the soil. So this is a really difficult process to measure because we have so much of that nitrogen gas in the atmosphere. So it's really hard to measure a small increase into the atmosphere. But we do know that it is the only known source of a sink of nitrous oxide in the soil. But it's quite largely unknown how plantain affects that process, and it could be that explains again some of the variability we're seeing in our soil in our results. So, in conclusion, coming back to the 4 potential mechanisms that I talked about before, and as I said, mechanism 1 I didn't touch on in here, but in our studies we found no evidence that mechanism 2 happens, so we didn't find any evidence that there is the BNI compounds that animals might ingest them through taking plantain in the diet and then excrete them with their urine. But we definitely find evidence of mechanisms 3 and 4 where plantain compounds could be into the root exudates, or plantain could change the microclimate and the N uptake in the systems. We don't know for sure yet whether it's one or the 2 or both, but we do find there is certainly a plant effect with quite a lot of variability in the results to date. And some of that variability that the results to date have pointed at could be the plant N uptake. As I showed in one of their experiments, there was quite a lot of difference in N uptake. There could be an effect of N loading, as our data analysis showed, and there could be pointing again to mechanism 1, where if you have plantain with a reduced N loading, that could have an effect, and that might be in a positive effect that we might see in the results. There's definitely an effect of some soil physical characteristics, and the field capacity of the underlying soil type that we're using seem to have an effect, again pointing to mechanisms 3 and 4. And then the final big question that we have is around complete denitrification and the effect of plantain on N2 emissions. And all of these, really, we need to really start getting a better knowledge of the fundamental mechanisms and understanding of what drives the mechanisms of plantain to reduce nitrous oxide emissions. And some of the key next steps that might help to point in that direction is really to start using our knowledge that we have to date and build it into refining our process-based models, and to include these plantain. And I particularly put it the way I put it with plantain effects, because I think it's not we can also look at plant effects as a whole and use plantain and the knowledge we have gained to date as a model plant to see how plants in general and what plant traits can affect nitrous oxide emissions. And then we, based on that modeling and do some scenario testing, we then can really start to hone in on what are the fundamental research gaps that we have, and how can we conduct some controlled experiment to test these hypotheses in more detail. And with this, I'd just like to acknowledge the funding of the Egg Emission Centre, and also the New Zealand government through the GRA has funded part of this study. And thank you to, again, Dairy NZ and the Plantain Potency and Practice Program for access to the less limited trials. And thank you for your attention.
Super. Thanks, Cecile. And look, can I remind everybody, we will have questions and answers at the end of Dominica's talk. So please don't be shy. Use the questions and answers tab at the bottom of your screen. Type them in. If the questions can be quickly answered, we'll probably answer them online. But then some of the other questions we will use within the discussion session. And you can also vote for your favorite questions if you want. So please don't be shy. Use the questions and answers tab there. And just a reminder that the webinar is being recorded, and that recording will be made available later. So now over to you, Dominica.
Thank you very much, Carl. So I will share my screen. If you could just let me know that you can see it and it's okay, that would be perfect.
Yeah, perfect.
Super. Okay. So good morning, everyone. Thank you so much for the opportunity to talk to you this morning about the Irish experience with plantain, but also a little bit kind of beyond plantain. So Cecile set the scene for me beautifully, especially kind of going into the details of those different mechanisms that plantain might or might not reduce emissions by. So I'll go from a slightly different perspective and just give you a bit of a pit stop tour of what we've been doing in Ireland. So bear with me, it is a little bit different. So before I move on to my slides, I just want to acknowledge all my colleagues and technical help that, that without it, all that work wouldn't have been possible, and very gratefully acknowledge all the funding that we've received for the previous and ongoing work that I'll mention in my slides. So first of all, Ireland is not dissimilar to New Zealand when it comes to our emissions. Ireland produces about 55 million tonnes of CO2 equivalents a year, and nearly 38% comes from agriculture. So similar to New Zealand, it just shows the importance of the sector, but it also means we are the biggest, sector producing greenhouse gases in Ireland, and there is a need for mitigation. We are also legally bound by targets to reduce emissions, and they are quite challenging. When it comes to agriculture, similar to the one in New Zealand, most of emissions come from methane, so enteric methane, but we also have some manure methane. 22% comes from nitrous oxide and a couple of percent from carbon dioxide. So most of carbon dioxide emissions are actually counted under land use change and forestry. So they're not they don't come into agriculture, kind of out of scope of today's talk. Then when it comes to nitrous oxide, the largest source of nitrous oxide in Ireland is from synthetic fertilizers. And then the second one is animal excreta deposited at pasture. We also have some emissions from manure application and from cultivation of organic soils. So this is, I suppose, where our agriculture starts to differ, where we typically house animals indoor over winter. And that means they produce, they defecate and urinate in the animals' houses. So we then store and spread that resulting manure when conditions become more favourable. So that's that part of the emissions. Then when it comes to research in Ireland, our scope is kind of beyond plantain. We're very much interested in plantain and its possibilities to reduce emissions of nitrous oxide and potentially have other benefits. But very much so, we're looking at what's called mixed species swards that typically include 3 functional groups. So grasses, clovers, and forbs or herbs. And that's where plantain comes in. So I suppose we're trying to look at it from kind of very holistic perspective. We work at different scales, starting from kind of very small scale, even smaller than lysimeter, but I kind of mentioned lysimeter here, plot and field scale, and we also have a few examples of kind of a system scale, farm scale research. And we're interested in a number of responses. So beyond greenhouse gas emissions, also plant productivity, animal performance, nitrate leaching, biodiversity, climate adaptation. And we're looking at responses from a variety of nitrogen inputs. So as I said, synthetic fertilizer, animal manures, animal excreta, and also AD digestate. So anaerobic digestion is a process and it's processing that's quite popular in Europe at the moment, and it's gaining popularity in Ireland. So this is quite a new branch for us, and a new kind of type of manure that might end up being applied to agricultural soils. So starting with nitrous oxide emissions from synthetic fertilizer and the impact of the sward. Here is an example of previous work from Saoirse Cummins. right, what this is, I want to spend a couple of minutes just explaining what this diagram shows, because I appreciate it might be very new for people. This is what we called a ternary diagram, where you can see that each corner of that triangle represents 100% monoculture of a certain species or functional group. So on top we have grass, on the left clover, and on the right herb. And then anything within that triangle, if you picked a point on it, would give you a combination percentage of the three functional groups. So in a way, this works similar to a soil texture triangle, if you're familiar with it. We use it for experiments where we used what's called simplex design. It's just a type of experimental design, very popular in, or very common in pharmaceutical studies, and it gives a possibility of really high statistical power while having a large combination of different grass species here. What you can also see is shading. So the colors represent the strength of a given response. So you can put a lot of different responses on this graph. The lightest shading is the minimum of the response, and the darkest shade is the maximum of the response. So there's a scale there at the bottom to read, and I'll be showing you a few of those graphs. That's why I'm going into details of how to read this graph. So what you see here is N2O, so nitrous oxide intensity, which is nitrous oxide emissions scaled to a pasture yield. And in this particular work, we saw no effect of grassland composition on N2O emissions alone. However, when you take into account pasture productivity, we saw that when we scaled nitrous oxide emissions to dry matter yield, emissions were reduced by 24% from a 6-species mixture grassland, which contained 2 grasses, 2 clovers, and 2 herbs, those herbs being plantain and chicory. So that response was 24% lower compared to a ryegrass monoculture, both receiving 150 kilograms of nitrogen. Then when you look at emissions scaled by nitrogen yield, the mixed species swards actually reduced that emission intensity by over 40%. So not necessarily emissions on its own, but when we when we take into account pasture productivity, this is quite significant. So that's just an example of I'll just give you some examples of studies today. Another one that we've looked on N2O from synthetic fertilizer applied to those different swards is we looked at potential pathways of production of N2O. So what you see here is a molecule of nitrous oxide, which has 2 nitrogens, and depending on the typical the kind of signature of nitrogen within that molecule, we can then look at, differentiate emissions coming from nitrification or denitrification. And this is called site preference, okay? And the low site preference denotes denitrification, and high site preference indicates nitrification. And we looked at that response in low and high soil moisture conditions. So in dry conditions, what you see here is kind of the maximum response pointing towards herb, particularly plantain. So if nitrous oxide comes from nitrification in this case, then if we have plantain in the swards, it means with its BNI traits, it might potentially reduce emissions. In the situation of wet soils, we see situation shifting somehow with the site preference, the top of site preference kind of somewhere in the middle, so for mixed swards, and it's a kind of wide space for mixed swards. But we also have that lowest site preference indicating denitrification in the corner with high proportion of legumes. We have seen in our previous work that legumes might be associated with higher nitrous oxide emissions because of their potential to fix nitrogen. So the combination of high legume proportion and N2O coming from denitrification might mean higher emissions. Moving on here, this is my last slide on the synthetic fertilizer, but what I want to show you is I mentioned that simplex design that we've used before. It employs quite a few plots. So we usually have all those plots receiving the same rate of nitrogen, and maybe have just perennial ryegrass monoculture receiving 0 nitrogen as a control. But here for the first time, we have really large amount of plots. And then, so this is an ongoing work and a joint between researchers in the Republic of Ireland and North of Ireland, Northern Ireland, where they carried out this experiment in 2 sites. And here you see an average of the emissions from the 2 sites. And you see we have results for the full simplex design for 0 nitrogen and 150 kilograms of nitrogen. So for the first time, we will be able to use this type of experimental design to quantify nitrous oxide emission factors. So this is really exciting for us, for me anyway, particularly because for the first time we have the opportunity to explore emission factors from a wider variety of grassland mixtures, and it's something kind of very new that we've never done before. So I'm looking for the conclusion of this work. But now I want to move on to urine nitrogen. So I can, Cecile again, showed a lot of work that was done in New Zealand. So we're, I don't want to say we're behind, but we are looking at the impact, similar impacts in Irish soils. And so far we can also see as you can see in the graphs here, we see an increase in urine volume when the grassland swards include plantain over just perennial ryegrass monoculture or a grass clover sward. So the graph on the left-hand side, you see clear increase in urine volume. In the middle, you see associated with that volume increase, there is a decrease in urine nitrogen concentration. And then on the right-hand side, it's a combination of those 2 metrics, where you see clearly that the lowest urinary nitrogen excretion is in those swards that contain plantain. I will mention here that we had a high inclusion rate for plantain of 50%, but that's kind of, to give the best chance for plantain to actually see this response before we delve deeper into it. So what that urinary N means for emissions. Here, another ongoing work, you see results from ammonia emissions on the left-hand side and nitrous oxide emissions on the right-hand side. We applied we applied 3 rates of urine N concentration, resulting in 3 nitrogen loadings at low, medium, and high, and also a 0, which was the water control. And we applied those treatments to 3 different sward types, again, grass clover, and multi-species, so the 6 species mixture that I mentioned before. So there was plantain, there was also chicory. And first of all, you see that lowering concentration in nitrogen urine mitigates both emissions of nitrous oxide and ammonia. But additionally, you can see that emissions are lower from urine deposited in multi-species swards compared to less diverse swards. So again, we're kind of alluding to those different mechanisms of how plantain might reduce emissions. We also have some ongoing kind of emerging research where we can't really share results yet, but I do want to just point you out to what we're doing. So in this particular project, BNI Pastures, that Cecile is a part of, and colleagues from Teagasc, they're delving more into the impact of plantain on urine characteristics and urination events, and the impact of season, soil, and kind of what impact it has on patch size and on emissions, with the view of upscaling and modeling those emissions. Then I think it's in the same project. It's an very interesting development that, you know, we've been looking into plantain now, but the question is, well, there might be other species that we're a little bit more familiar with, that like perennial ryegrass, that might perhaps have BI nitrates that could be useful in reducing future emissions. So there is currently screening work ongoing, looking at different varieties of perennial ryegrass, looking for those beneficial BI nitrates. Then I'm also a part of another joint Irish-New Zealand projects with colleagues here in BSI. Carol and myself are actually visiting at the moment, so we're on the same time zone, which is very useful. But in this work, where we will be investigating nitrous oxide emissions from animal urine deposited to those different sward types, and potentially manipulating urine N content as well, to measure emissions and then employ statistical and process-based models to model those emissions. And the ultimate aim of this work is to try come up with a new method of quantifying dynamic nitrous oxide emission factors that can be used by greenhouse gas inventory compilers. The usefulness of this would be that we would be able to more accurately account for emissions, but also for mitigation efforts. So again, very exciting work. Now, I don't have much time, so I don't want to go into this really big detailed table here, but this is just a flag because I've mentioned AD digestate at the very beginning. So just wanted to say that we are looking into both, you know, growing the various grassland communities to use in AD processing, but also then investigating emissions associated with recycling of resulting manure back onto grasslands. So there is work there being carried out, and so far comparing digestate to synthetic fertilizer, we see that emission intensity, that emissions are lower from digestate compared to synthetic fertilizer, but there is also an impact of sward composition, where the high diversity swards have lower emission intensity compared to grasslands. Then again, just to point, I know this isn't nitrous oxide, but nitrate leaching is, is a major concern in both countries and internationally. And in recently published work, we've seen that in both well and poorly drained soils, when grassland, which is a monoculture perennial ryegrass, when it gets supplemented with either 30 or 50% plantain, it has a potential to reduce nitrate leaching, which we can see in both years in well-drained soil, which is a Clonroach soil here. And while we don't see this impact in the poorly drained soil in the first year, we do see a reduction in the second year. So once the plantain is well established, we can see a beneficial impact on reducing nitrate leaching. And similarly, work conducted by colleagues in Teagasc and Moorpark has also seen a reduction of approximately 20% of total nitrogen leached following urine deposition to 2 soil types, and that's regardless of the soil type. And I also mentioned to you at the beginning that when we're going through the different scales of research, we do have some system effects research. So we have trials going on experimental farms in Teagasc and colleagues in UCD, which are looking at systems such as beef, dairy, and sheep. And just the example here of our own 2 farms. So in Teagasc, we run 2 systems, which one is kind of your typical perennial ryegrass clover at receiving high nitrogen, and multi-species with the 6 species mixtures which receive much lower inputs. And what we see that the outputs are not reduced in terms of annual milk production, milk solids, and also pasture productivity. Similarly, colleagues from Moore Park run tree systems with either perennial ryegrass monoculture at very high nitrogen, a perennial ryegrass with clover or multi-species at half of that nitrogen input. And similarly, they are not seeing any negative impacts. So I'm going to round up now. Just very quick conclusions is that what we've seen so far is multi-species swords can reduce nitrous oxide emission intensity. There is a broad safe operating space for mixtures. So what you have maybe seen on those ternary diagrams is that there's no kind of a small circle of that, those low emissions or emissions intensity. There's kind of a big area of lighter color of response. So that kind of shows that safe operating space. So it doesn't mean that you have to get it exactly right with the proportions that we use in our trials, because you will still get benefit if you have those key species. And plantain, we know so far is the key species in those swords. And plantain containing swords reduce soil nitrate as well. And we have also seen in our work enhance yields under water stress conditions. And we see grassland composition manipulation as a practical farm scale action that we already see farmers take up, and our Department of Agriculture supporting them in doing so. And we do have quite a lot of ongoing research in this space. So in the future, we will be able to report on more of our results. So thank you very much. And again, just wanted to acknowledge our funders and our collaborators and colleagues that made this work possible. So thank you very much.
Okay, thanks, Dominique, and thanks, Cecile. And look, there's quite a number of questions in the questions and answers. But look, I think you've both given a really comprehensive overview of the ongoing research. And I suppose looking from the outside in, it's really complicated. Cecile, you have 4 different mechanisms, and you presented each of them separately, but they're all interacting with each other. So it is a difficult space, and difficult one for maybe some of our farmer viewers to watch and to really distill out what does this mean for them. And I suppose at a high level, what I'm taking from it is, look, having plantain in there is a good thing. From an agronomic perspective, it can be really beneficial. I think from a nutrient and all of those other perspectives, it can be really useful. And there are indications coming from the wealth of research across both countries, showing that there are positive benefits on nitrous oxide, but also on other sustainability responses, be it nitrate leaching or ammonia and things like that. So again, a lot done, still a lot more to do. So look, what we might do is start to probe into some of the questions. Cecile and Dominique, you'll see them in the Q&A tab there. So look, I'll try and pick out a few to focus on, but if there's ones in there that you're particularly keen to get your teeth into, let me know. And don't forget, audience, please keep putting in your questions. We'll do our best to answer them in the next sort of 10 to 15 minutes or so, and those that we don't answer, we can answer online, I think, anyway. So look, the top one that I have in mind is around when cows are fed plantain, they excrete BNI. We know that nitrogen doesn't just disappear. Where does the nitrogen end up? So maybe that's something both of you could contemplate.
Yeah, I'll make a start. Thanks for the question. It's an interesting one. I might make a slight nuance on it, because our research is not necessarily showing that they do excrete BNI, but the plants themselves may excrete BNI into the soil. So it's slightly different as what you describe it here. But you're right. I mean, there is, if the N is not lost, where does it end up? And some of the work that I showed is that there is an increase in nitrogen uptake. So that's definitely one mechanism. The other is it stays in the soil as ammonium longer if we inhibit nitrification, and ammonium is just less mobile than nitrate, so it can be utilized over a longer period by the grass as it grows. So I think ultimately that's where it may end up. Or, as I said before, and that's the big unknown, it may end up as N2 gas in the atmosphere, but we don't know that yet.
Okay. Thank you. Dominique, I don't know if you have anything to add. If you don't, we can move on.
Very, very quickly, Carl, maybe just before that, that nitrogen is excreted in urine when animals graze on swards containing plantain. There is, because there is less nitrogen deposited or excreted in urine, it's actually a shift in partitioning. So there's more nitrogen being transferred to dung, which is more stable organic. And so again, emissions from dung are typically much lower than from urine.
Okay. Another couple of questions here again. I think some of the questions are quite short, so we might try and type those in online. So there's one here for you, Dominica. I have a question related to your results so far that a single default grazing excrete emission factor is no longer appropriate and that Swartz specific factors may be required. So yeah, so should we be looking at I suppose refining inventories based on the sward type, nitrogen form? You
So
might
that
speak
is
about that.
that is exactly what we're trying to do in the new project with colleagues from New Zealand here, Dream Factors, where we will be applying cattle urine to those different swards and measuring emissions and then scaling up to modelling. So that is the view, and kind of seeing the differential in emission profile from synthetic fertilizers applied to different swards. Yes, I would say we are hoping to move in that direction of sward specific emission factor.
Super. There's a couple of questions here which I might try and join together, which is around, I suppose, the phytochemicals or the impact that plantain and other species in the multi-species might have around animal performance, milk or meat output, or any health issues. So are there, while they're not directly N2O related, are you aware of any of the other positive effects of these species?
It is indeed not part of what I'm intimately involved with the results, but the plantain practice and potency and practice program that I mentioned before has looked at the impact on milk particularly, and I believe there is no impact or no negative impact of including plantain, and there seem to be no yield penalties either. So whether it's going the other way, that's actually positive, I'm not entirely sure, but that's what I've read and seen.
Okay.
And that is similar in Ireland in terms of what we see in our research farms, that there's the yield, the milk yield and the milk solids are very similar. Again, also not my area of research, but plantain has been used for animal health. Like I think is it anti-warming or warming? Yeah.
Yeah. No, there certainly seems to be some health benefits associated with plantain and other herbs. So just moving on, actually, Dominic, you skipped across one comment, which was around at the system scale. And this links back to one of the questions, which was around, I suppose, milk and meat output. So you showed a systems project in Johnstown Castle where you've, quite dramatic different nitrogen inputs and yet very
Yes,
similar
yeah, and
performance.
the same performance. Yes, and that's a study that's been ongoing for a number of years now, with colleagues there, and so far like these are the outputs. Yeah.
Exactly. And again, that sort of highlights that interconnected nature of all these potential mechanisms, but also some of the responses that a farmer might do. The cost of adding plantain, well, can you reduce your fertilizer inputs, where you're introducing plantain, clover, and chicory? So like it would indicate that potentially you could. Sorry, it's hard to moderate this and go through the questions at the same time. So there was a very quick one, Dominica. Did you look at chicory alone in the Irish study?
So we would have had a chicory monoculture in the within the simplex design, but apologies, I cannot recall the exact results now from it.
Okay, but look, the reference
Yeah.
was on the graph, so
It is, yeah.
the viewer can go and have a look at the paper
Yes.
or email you afterwards if they want to know
Yeah.
more. There's a question here around would swards composed of multiple ryegrass varieties provide similar reductions to the multi-species swards, or potentially ryegrass and plantain, which again sort of links to maybe some of that ongoing work, Cecile and Dominique.
Yeah.
Cecile, maybe
Yeah.
you could say something about the ongoing work around ryegrass and BNI.
Yeah. As Dominica showed, there is work in ryegrass, and that is happening in New Zealand as well. And New Zealand and Ireland are working together in this. And in the BNI pastures program, we're looking at the ability of ryegrass to induce nitrification inhibition and affect BNI. And there's certainly some evidence that that might happen. There are certain cultivars, even under the elite cultivars, that seem to be having an effect. So that is definitely ongoing work. And that study is also looking at interaction between ryegrass and plantain, and using high and low BNI species of each and combinations to see if it's a multiplier effect or how they interact. So that is future work that is ongoing.
Okay. Sorry, I'm trying to read multiple things. Dominique, do you want to say anything on that yourself?
No, I think we've so many questions we need to try answer more. I'm looking through the questions here as well.
Thank you. Look, there is quite a long one. So I'm going to try and summarize it, but it's around blaming animals as a significant source of N2O emissions when it's also emitted from ungrazed and unfertilized grasslands and even by native woody vegetation before land reclamation. So I'm not quite exactly, I don't think anybody's blaming animals here.
No,
what we're trying to
I
do
was
is
just
to
going to say
better
that,
understand what's
Carl,
happening.
we're not blaming animals for anything. It is what happens. I mean, we know that there is a lot of nitrogen that is returned in the system in very small concentrated patches, and that is what it is. And we're trying to measure and trying to minimize the emissions or the losses of that nitrogen from those patches.
And maybe just to add to it from Irish perspective, our biggest source of N2O is actually synthetic fertilizer. So while, animal urine is very significant, synthetic fertilizer is the single largest one. So that is added nitrogen. That's nitrogen fertilizer that usually has a very high carbon footprint, and it's fixed from the air. So we're taking the nitrogen that was benign in the atmosphere as dinitrogen and fixing into a reactive form of nitrogen that we can then use for agricultural purposes, for productivity. But at the same time, we are adding reactive nitrogen into the system. So what we've shown here through our research is, we are trying to actively reduce that fertilization levels by kind of nature-based solutions such as mixed species swords.
Perfect. Look, moving on. So I'm not quite sure what varieties of either plantain or multi-species that you look at, but how important do you think cultivar to cultivar differences might be within plantain or different elements of multi-species swords?
So well, I can pick that up, Dominica, if that's okay.
If you want to start,
yeah. So there are differences, a potential difference between cultivars, and there has been work done in New Zealand to screen different cultivars of plantain for its ability to induce BNI. And from that original screening, we have then subsequently taken some of the higher, medium, and low BNI species out into the field and done some experiments there. And yeah, it does seem to be that there is a difference in the different cultivars. So unfortunately, we didn't know exactly what the cultivars were. We only had them classified as high, low, and a medium BNI. So, but there was differences indeed between cultivars.
Okay, super. And look,
They
potential there then to obviously breed for not just nitrous oxide. Like one of the biggest challenges we have with herbs is trying to keep them in the sward.
persist.
Absolutely.
Yeah.
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 So one of the key findings is yes, plantain doesn't maintain in the sward as long as ryegrass would. So there are things that you need to do to keep it in the system. Yeah.
Okay, we're nearly there
And
too. Go on,
we're
Dominica.
we actually have started a breeding program in Teagasc looking into cultivars, kind of breeding for persistence. So fingers crossed there's some
Brilliant.
improvements to happen in the future.
Yeah.
That's great. And look,
So,
final two questions. If what do you both see as sort of the key research gaps or the key research challenges that this research is facing? If you had $1 million tomorrow, where would you put
mm,
it?
mm. Yeah, I made a start on that in my last slide, I suppose. Look,
Mhm.
what I keep saying is if we want to use it as an anti-mitigation technology, and we've started measuring the nitrous oxide emission reductions, if we had found positive results all the time, then I wouldn't have worried about what the mechanisms are exactly. We could have started using the knowledge we had into our inventory and start giving people credit for using plantain as an anti. But the reality is there's so much variability that we now need to start really understanding and delving into the detail about what it is that drives that variability and in what circumstances we get those high emission reductions as we do find sometimes because they are big. So we can maximize those. If the potential is so great that I think we need to start looking into the detail and it is understanding about the mechanisms and when does it work.
Very good. Thanks, Cecile. Dominica, do you
I'll,
have
I'll
anything to
leave
add?
it.
Even
I'll
that's
leave it at
fine.
that.
Okay. So look, your parting comment, if you have one sort of practical thing to say to a farmer around either plantain or multi-species swards, what would you tell them, or what would you say to them?
I think that in the climate that we're in, and farmers are under so many pressures, facing reductions in fertilizer inputs or potentially reductions in animal numbers, this might actually be a really promising option compared to other ones. And, even though there are questions over persistency or how well to manage those swords, it is beneficial to just take the first steps and start trying.
Okay, Cecile.
Yeah, I'll second that. And I
Okay.
think it is really thinking about the goals and what you're trying to achieve. And if nitrogen reduction is part of that, then certainly consider it as one of the tools in the toolbox.
Cool. And I presume there are probably support services in New Zealand and Ireland helping farmers to establish and manage and maintain those swords. And again, probably not putting all the eggs in one basket, but try it out on a paddock, see how you get on.
Yeah.
And reach out and use those supports. Okay, look, with that, I'll bring the discussion to an end and want to thank both of you for your very interesting talks. And I've certainly learned a lot, and hopefully the viewers have also. And obviously, they can reach out to you through the internet if they have further questions. And a copy of the presentation will be up online in the next week.
Great.
So,
Thank you
you
very
very
much.
much.
Thank you. Thank you.